Hello there. My name is Michael, and I would like to personally thank you for taking the time to watch the video presentation on how my master's thesis would have been performed if not for the interruption of the COVID-19 pandemic. As you can see here, the original study was to be called Analysis of Brain Activity During Aquatic Walking in People Post-Stroke. But, for the purposes of this video, we shall be foregoing the aquatic portion of the study and will be focusing on the brain activity and walking aspect of the study. Before we continue, though, I would like to give many thanks and recognition to my committee members, Dr. Hinkle-Lipsker and Dr. Furtado, and my committee chair, Dr. Jung, for helping me create and design this study. They have provided me with invaluable guidance, and I would not be here today without their help. Speaking of people who helped me along the way, here is my grandfather, seen holding me back in the long time ago of 1992. About 5 years ago now, my grandfather suffered a severe stroke. While he is still highly ambulatory and mostly independent, cognitive deficiencies and difficulties with motor skills have become more prevalent and more pronounced in recent years, thus inspiring me to pursue a deeper dive into the cognitive effects of the post-stroke brain. With this curiosity and the guidance of my instructors here at CSUN, I began to research and discovered articles such as these.
In my research, I found that strokes occur 795,000 times in a year in the United States, or about once every 40 seconds. The Mayo Clinic defined stroke as an occurrence when the blood supply to part of the brain is interrupted or reduced, preventing brain tissue from getting oxygen and nutrients. These strokes can occur in two forms: ischemic, where blood in the brain is blocked, such as by a clot, and hemorrhagic, where a blood vessel bursts and the brain bleeds. It is possible that ischemic strokes may cause bursts in the brain's blood vessels, leading to hemorrhagic strokes also. The combined severity and prevalence of strokes cause strokes to be the leading cause of long-term disability in the United States. So, by falling down this research rabbit hole, I discovered articles pertaining to my interests outside of stroke, such as aquatic activity and brain functionality. The first one is the efficacy of aquatic treadmill training on gait symmetry and balance in subacute stroke patients. This was a 2017 study by Lee and colleagues. It was an intervention study that looked at 21 individuals over a 15-week period. They wanted to test the efficacy of aquatic treadmill training for individuals who had previously experienced a stroke. As a result of the aquatic treadmill training, the researchers discovered that gait symmetry and balance both improved. Gait symmetry was analyzed using a motion analysis system, and balance was measured using the Berg Balance Scale and the Activity-Specific Balance Confidence Scale. Another one of these studies I looked at was the 2018 study, The Effect of Aquatic Treadmill on Clinical Function in Stroke Patients, by a different Lee and colleagues. So, once again, they were looking at 30 post-stroke individuals in an intervention study. The purpose of this test was to gauge the effectiveness of aquatic treadmill training on lower limbs. This study found that aquatic treadmill training increased the strength in the lower limbs of the participants. The strength in the lower limbs was measured with asymmetric contraction holds to test the amount of force output they could find in the limbs of participants. Ultimately, these two studies agree that aquatic treadmill training is beneficial for individuals post-stroke. So, as I mentioned earlier, I also had a piqued interest in brain functionality, more specifically, the brain's response to a stimuli. Therefore, with the brain being the major organ involved with stroke, I researched literature on brain response in aquatic walking, which led me to the article by Parfit and colleagues. This study, Cerebral Blood Flow Responses to Aquatic Treadmill Exercise, was a cross-sectional study which compared brain and heart rate responses between land and aquatic treadmill exercise in 11 healthy individuals. What this study was looking at was individuals at varying depths and how the brain and heart would respond, comparing those results to the individual's land-based exercise readings. In conclusion, they found that brain blood velocity increased with the aquatic walking, while aquatic walking also caused a decrease in heart rate. They discussed that these findings can promote adaptations in individuals such as healthier heart rate and healthier brain activity by training with the aquatic treadmill. The cross-sectional study I looked at by Pugh and colleagues, the impact of water immersion during exercise on cerebral perfusion, again looked at healthy participants, in this case 15. They wanted to compare cerebral perfusion between land and aquatic exercise. Cerebral perfusion is defined as the amount of oxygen used and distributed around the brain during the aquatic exercises. It was similarly found that the oxygen consumption increased in participants' brain during aquatic exercises. So, according to these studies, cerebral blood flow increases in water, and aquatic treadmill training appears beneficial to participants.
So, overall, with these articles, it can be safely assumed that cerebral blood flow increases during aquatic activity, aquatic treadmill training is beneficial to participants who use it, and using an aquatic treadmill promotes a lower heart rate response during physical activity. What has yet to be studied, though, is the brain's response to an individual post-stroke during gait training in differing walking conditions. Therefore, I came up with a question. Is there significant difference in brain activity for people post-stroke during different walking conditions? And to answer that question, I have designed this study with the purpose of comparing brain activity in the prefrontal cortex of post-stroke individuals during different walking conditions. And for this study, my hypothesis is that there will indeed be a significant difference in brain activity across different walking conditions for people post-stroke.
It is here at the Center of Achievement, on the campus of California State University Northridge, that this research would have occurred. Prior to the pandemic, we had planned to use these aquatic treadmills seen here and this main therapy pool in order to perform this half of the study. This is the fNIRS NIRX Sport. This device measures oxygen saturation in parts of the brain using infrared light near the surfaces of the scalp. First, the fNIRS needs to be checked for calibration. The calibration is performed using the electrodes and this rectangular box here. After everything is connected, insert the electrodes into the holes in the following pattern. Once the pattern is completed, turn on the fNIRS to ensure that all electrodes light up. Once all electrodes react to the fNIRS, the device is calibrated. After the machine is calibrated, the next step is to measure the head of the participant to ensure the best fit of the equipment for the best possible readings. Using a tape measure, measure the participant's head in centimeters, making sure to pass through the nasion and anion points. When measuring, be sure to round to the nearest whole centimeter to get the correct cap size. After getting the correct sized cap, place the cap atop the head of the participant and attach the electrodes into the correct area. The electrode placement will change depending on the area of the brain being observed. For our purposes, we will be measuring the prefrontal cortex. Ensure that the wires are attached in such a way that they avoid the participant's eyes and run down the back of the participant's head. You can use a probe tool to ensure that the optodes have good connection to the scalp by moving hair out of the way. You can stabilize the cables with the provided Velcro straps. It is still recommended to use the overcap even if the main cap is not loose, as it will help the optodes achieve better readings. Do your best to keep participants comfortable, as this cap is often very tight and uncomfortable. Once the cap is on, use the computer to make sure the optodes have a good connection to the scalp of the participant. You can open NearStar and use the Configure Hardware tab and select Calibrate Signal. Channels will show colors depending on how good the connection is. Green and yellow equals good, red equals okay, while white is bad. Use the probe to again adjust the red and white zones as necessary. When satisfied with the setup, you may then begin data collection. The participant will begin with a short rest prior to any activity. This will allow the fNIRS to gather a baseline for the participant at rest. Next, the participant will stand on the Biodex gait trainer with their legs spread. This will stop the participant from suddenly jolting when the machine is turned on. The participant will go through a quiet standing period of 10 seconds to allow their brain activity to return to baseline. Once ready to walk, the participant will begin to walk on the gait trainer. The gait trainer will slowly accelerate to the participant's comfortable walking speed as determined from a previous visit. The acceleration time should take exactly 10 seconds. Once the participant reaches their comfortable walking speed, they will walk comfortably for another 10 seconds. Afterwards, the Biodex should decelerate to a complete stop for another 10 seconds. Repeat all these steps starting from quiet standing for 10 seconds 3 times to get 3 good readings on the participant. Note, the Biodex should be surrounded by white sheets to reduce the amount of outside influence to the participant senses. We are not doing that here for the sake of filming. For this portion, we will be using the hallway. The participant will once again be seated for setup of the fNIRS device. After receiving baseline data while sitting, the participant will then stand for 10 seconds quietly. The participant will then walk at their comfortable pace in a straight line for 30 seconds. After 30 seconds of walking, the participant will turn around 180 degrees and stand quietly for another 10 seconds. Afterwards, the participant will repeat the 30-second walking 2 more times. After the participant is done with the trials, it is now time to remove the device and begin extrapolating the data collected.
After data collection, the data from the fNIRS can be converted into a notepad file, which can then be converted into an Excel file. Right here, I have the Excel file for some sample data. Now, what I have here is a script detailing what I want the data to show. The first step is opening and organizing the data. If you double click on the file, you can take a look at the data. Right here, with this sample data, you can see all the measurements taken by the fNIRS. As you can tell, it's a lot. Over here in column V, we have time. All of these other numbers are data collected by the fNIRS. I have this script set up in such a way that will open up and automatically read the fNIRS data. And here, I have it set up to read 16 channels, or also 16 columns, because we only have 16 optodes on our fNIRS. So by clicking here, I can simply click Run and Advance. That will bring me to step 2, and you see right here on the right side of MATLAB that you have all of the data right there. If I didn't put these little semicolons in the script, you would also see it pop up down here, but we do not need all that extra processing. So here we want to filter the data through bandpass filters. I have the script running 16 times, once for every channel, so that way each channel gets filtered. Right here we have fs, which is our sampling frequency. The fNIRS that we have rates at 7.81 Hertz. So that's why our fs is set to 7.81. For our bandpass filters, we need a low cutoff and a high cutoff. As recommended by UCLA, I used 0.005 for our low cutoff and 0.5 for our high cutoff. Once again, looking at all 16 of these, they are set up in the same exact way. So simply hitting Run in Advance will move us to step 3. Step 3 is preparing the data to be graphically read. What these functions here are doing are taking every single one of the filtered data and squaring it and then square rooting it, so that way the data is then all positive. It does not make sense to have less than 0 oxygen in the brain. What I also have here is, for visual purposes, what I have here is an average of the first 12 seconds of data. That is the baseline data. What I am doing here is I am making it so that it is more easily recognizable on the graphs by changing the first 100 cells into the average. Now, normally this is frowned upon and heavily discouraged in data collection, but for the purposes of this video and to make it more easily seen, I'm doing it. Once again, simply clicking Run and Advance will bring us to the next step. As you can see, we have all of the new data here on the right side. Now it is time to give a visual representation of the data by plotting all of the data. Here we have each plot, all 16 channels, being plotted against time. Time will be the x-axis in seconds, and we have each channel as our y-axis. This part of the script will allow for 16 different graphs to show up all at once, showing a visual representation of the data. At the bottom of this step, I also have this G input function so that we can see exactly where the peak data is and click it. That way we get the X and the Y axis for the peak data. We also have onset time, which isn't exactly necessary since we are timing in seconds on the X axis, but onset time is usually used so that way you can find exactly where in the data recording a certain point of the graph happens. So by clicking Run in Advance again, we bring up all 16 graphs and this little crosshair. This crosshair is the G input function. Looking at these graphs, you can see that each one is different because each one is a different channel. Each one of these graphs is recording a different area in the prefrontal cortex. Because of the G input function, I can go ahead and find where the peaks are on the graphs to mark the highest data point of each graph. Now, if you can see at the beginning of the graph, there is a little flat line. That is the average that I gave earlier, which is our general baseline. And while it looks like this all happened all at once over an 8-minute period, because of the previous methods, you know when and where each trial is started. So therefore, you can use that information to cut out unnecessary parts of the graph. Returning to the main script, the other way to find peaks is by using the max function. I have this set up so that way, if I just hit Run in Advance, all of the peaks show up right here in the side. We can see the highest oxygen saturation for each optode, or each channel. Now, one more step, very simple, just cleaning up the data. Right here, we have a clear function, which gets rid of everything that you don't need. So one more Run in Advance, and now all you have are your peak data sets. So now you have every channel and the peak data that it gets to. And you already know your average and baseline and the times at which these occur. So, after watching this video, I hope it is now clear how this experiment and how these results could be applied to a post-stroke population. Before I go though, I would definitely like to thank once again my committee members, my cohort for helping me, everyone who helped me develop this project, my girlfriend who helped me record, and every single person who watches this. I would like to thank you for sitting through this as well. If you have any further questions, I will be happy to answer them. Thank you.